right, perfect. Good morning, everyone. My name is Janelle Tam, and I'm here to present to you exciting findings on the use of nanocrystalline cellulose, or NCC, as a novel, renewable antioxidant. But first up, let's put it in context. What are antioxidants, and why are they important? As you can see from this figure here, an excess of free radicals or highly reactive atoms in the body cause damage to cells and tissues, whether by dismemberment of peptides or unnatural cross-linking of DNA. The body's defense is antioxidants, compounds such as vitamin E that trap these free radicals and help prevent the damage from occurring. However, when the body undergoes severe oxidative stress, free radicals end up accumulating in the body, and this leads to diseases such as cancer, diabetes, and atherosclerosis. Antioxidant supplements now, by combating these free radical damage, can help to actually prevent disease, support medical treatment, and extend the survival of the terminally ill. As well, as you can see from this table here, has a number of other applications in the food industry as a preservative and in the cosmetics industry as an anti-aging ingredient. However, there are three problems with antioxidants today. The first is that synthetic antioxidants have recently been suspected to be cytotoxic. Butylated hydroxytoluene, for example, has been seen to possibly be a carcinogen. Secondly, the question is of water solubility, because low water solubility leads to low bioavailability, which means that these antioxidants are less available for the body to use. The third question, or problem, is deterioration over long-term storage, which can greatly diminish the effect or the effectiveness of antioxidants such as vitamin C and E, especially if they are to be incorporated into a product that will be shipped somewhere before use. Therefore, there is a need for a non-toxic, water-soluble, stable antioxidant. And that was the question that I was trying to solve with my project. So the first place, or a good place to look, I thought, was at cellulose, which is the most abundant biocompatible material on Earth. When you dissolve the amorphous regions of cellulose via a process called acid hydrolysis or polymer degradation via sulfuric acid, you get a nanoparticle known as nanocrystalline cellulose. And you can see the breakdown right here. As well, there's an AFM micrograph up here of this rod-like shaped nanoparticle, which is 5 to 20 nanometers in diameter and 200 to 400 nanometers in length. What is great about this is that it has a projected North American market of $250 million alone, and it's only been really recently discovered, so this will be a boost to Canada's struggling forestry industry. This has been extensively reported in the media. As well, Domtar and FP Innovations, which are governments funded, actually recently opened a $30 million pilot production plant in Windsor, Quebec of January of this year, and another is scheduled to open in Alberta. Furthermore, it is renewable, abundant, stable, water-soluble, and non-toxic, which means it perfectly fit the requirements for the antioxidant that I was looking for. Most importantly, it has an abundance of hydroxyl groups on its surface, which have been linked via research to antioxidant properties, and which also allow nanocrystalline cellulose to be modified via, say, the grafting of a strong radical scavenger onto its surface to enhance any existing antioxidant properties that it may already have. Buckminster fullerenes is one such radical scavenger. It is a hollow sphere of carbon that is already commercially used in a lot of cosmetic formulations for its antioxidant properties to reverse aging. An example of this is vitamin C60, which is a product out in the market right now, which is basically polymer-wrapped fullerenes. It is insoluble in water, which is the only obstacle, but that can be modified. It can be changed to become water-soluble. And so, I had two main objectives to my project. The first was to create water-soluble fullerenes and then graft it onto the surface of NCC. The second was to compare then the antioxidant properties of just NCC, this new NCC fullerene complex I had synthesized, and other similar synthetic polymer fullerene complexes specifically selected to represent commercially available products. My hypothesis was that because free radicals are reduced by the addition or donation of a proton or electron, that NCC would be a strong radical scavenger due to the abundance of hydroxyl groups on its surface, which can then donate a proton and reduce these radicals to their stable state. The second conjecture was that the NCC fullerene complex would be an even stronger antioxidant because Buckminster fullerenes are already very good radical scavengers. I had three phases to my procedure. In the first one, I had to establish a protocol for testing antioxidant properties because there are lots of different ways you can do it out there. And I selected the DPPH method, which is a pretty standard test that is used in a lot of research. The 2,2-diphenyl-1-pickyl hydrazole is a stable free radical that actually displays a strong absorption maximum at 517 nanometers and a purple color when in solution. As DPPH radicals are reduced to a stable state, it goes from purple to yellow, and the peak here goes to a straight line. And this color change here is stoichiometric and thus can be monitored via ultraviolet visible or UV-vis spectroscopy. And that is how I monitored the radical scavenging reaction over time. So I can analyze its kinetics and also the overall inhibition activity. However, I encountered problems with protocol when I first started. I used the solvent DMSO or dimethyl sulfoxide, but I didn't realize that it actually had some radical scavenging properties. And so I only found this out after about 3 months of my research, which meant that 3 months of my research had been invalidated, unfortunately. So what I had to do was I had to quickly scramble and find a new protocol, which I did after extensive literature search. So you can see in this table here, I based it off of Yamaguchi et al., who was the original creator of the DPPH method. I used alcohol reagents as my solvents, and I basically customized it to fit my system specifically. I ran 3 control experiments just to make sure that I wouldn't have to go through that again and that my results this time would be validated. The first thing I did was I ran an experiment with ascorbic acid and matched that to literature data. The second thing I did was experimentally derive a DPPH calibration curve and extinction coefficient, and again match that to already existing research. And the final control experiment I did was a solvents control experiment to ensure that my solvent had no effect on my results. In the second phase, I synthesized the NCC fullerene complex in 2 steps. First, I created a water-soluble fullerene, as you can see here, and then I grafted it onto the surface of NCC. So for the creation of water-soluble fullerenes, what I did was I created saturated solutions of beta-cyclodextran in water and saturated solutions of fullerene in toluene, because they're both soluble in those solvents. I mixed them at 25 degrees Celsius for 2 weeks. And what happens is that in this solvent exchange reaction, the fullerenes in the toluene phase are encapsulated by the beta-cyclodextran molecules in the aqueous phase, and by this encapsulation via the host-guest complex, then you create a water-soluble fullerene. I then dialyze this for 2 days to remove excess reactants. In the second part of this, I conjugated my fullerene to NCC. So I did this by mixing beta-cyclodextran fullerene complex I had originally created in the previous step with 2.5 weight % NCC solution in water and sodium persulfate. I bubbled it with argon for 1 hour, mixed it at 65 degrees Celsius for 2 days, and dialyzed it once again to remove excess reactants. And how this works is the sodium persulfate actually radicalizes the oxygen atoms that then react with the pi electrons on the surface of the fullerene to form a covalent bond and successfully graft the fullerenes onto the surface of NCC, thus successfully creating my complex. In the third phase, I compared the antioxidant properties using the protocol I'd established in phase 1, the DPPH method, to compare NCC, the NCC fullerene complex, and all the polymer fullerene complexes shown in Table 1. I specifically chose these complexes to be representative of commercially available products to provide a good basis for comparison, and I'll be referring to these by their short forms. I chose 2 parameters to test or to quantify antioxidant property. The first was rate constants, which is a measure of the efficiency or how fast an antioxidant can scavenge radicals, which is determined by the equation shown, where A infinity, AT, and A0 are the absorbances at infinite time, time t, and time 0, respectively. The second parameter I looked for, or I used, was inhibition activity, which is a measure of the effectiveness, or how many radicals an antioxidant can scavenge in a set amount of time, which is determined via the equation shown. Again, these are the absorbances at time 0 and infinite time. Infinite time measurements were taken 3 days after the experiment was completed, and all data was normalized for the same fullerene concentration to allow for comparison. And I selected these 2 parameters to provide a holistic view of antioxidant activity, because a good antioxidant is one that scavenges a large amount of radicals in a short amount of time. And that is one of the problems with vitamin C, for example, that although it's very fast, its capacity is quite limited, which is why you have to take supplements day after day. So now for my results. In the first phase, I found my control experiments revealed that the protocol I was using was in fact reliable. The ascorbic acid run did match literature data, as did my experimental calibration curve. The extinction coefficient was consistent with what had already been researched. So this allowed me to confirm that the samples I were using had not deteriorated. But also, to be careful, I prepared a new fresh stock solution of DPPH before each experiment. In my solvent control experiment, the standard deviation between the rate constants for the same experiment in different solvents was extremely low, was negligible, and so I was able to conclude that solvent has a negligible effect on results, or the solvents I was using. In the second phase, I found that in both complexes that I produced, it was a homogeneous brown-colored solution even after fullerenes had been removed, which led me to believe that fullerenes had successfully been incorporated onto the complexes. But to further confirm this, I took the UV-vis measurements of both complexes, and I found that there was a characteristic peak at 330 nanometers, which is characteristic of fullerenes, which provided further evidence for the success of the grafting reaction. In the third phase, when I was doing the kinetic analysis of my curves, I found something kind of unexpected, and I was confused at first. What it basically was a double exponential trend, as opposed to the single exponential trend that is usually seen and that I was expecting to see. And so after some thought and research and discussion, my hypothesis was that this is due to the presence of both unimers and micelles in solution. So as you can see here, these are the unimers and these are the micelles. And so my hypothesis was that because the polymer fullerene complexes are amphiphilic, and the critical, I used concentrations that were higher than the critical micelle concentrations, there were both unimers and micelles in solution. So what happened is that once the radicals enter the solution, it reacts first with the unimers because they're more accessible, which yields the first rate constant. And then after the free radicals diffuse into the hydrophobic micellar core, they then react with the fullerenes there, and which yields the second and slower rate constant, which you can see right here. To confirm this hypothesis, I prepared a purely unimeric solution and tested it and found that it indeed only had a single exponential trend. As well, NCC and NCC fullerene, which do not form micelles in solution, also had a single exponential trend. So I found this graph shows the rate constants of all the polymer or the complexes that I tested. What I found was that NCC actually was an excellent antioxidant that was superior to all the other polymer fullerene complexes that I tested, which I was really excited about. But what I found even more was that NCC, when enhanced with fullerenes, actually improved, so it became even better than it was before. This graph shows the inhibition activity of all of the fullerene, or the complexes that I tested. And I found NCC had a moderate inhibition activity compared to the rest of the polymers or the complexes. But as again, we see the same trend where the NCC, when enhanced with fullerenes, also improves, so inhibition activity increased. And so what I've found, or these results show, that NCC not only is an excellent antioxidant on its own, but it is very flexible, that it has the potential to be modified in various ways that can actually increase it and make it even better, which means it has huge potential for being an even more effective antioxidant than it already is. So in conclusion, I found that NCC does have antioxidant properties and is already better than polymer fullerene complexes. Secondly, I successfully synthesized the NCC fullerene complex and showed that it was more efficient than other polymer fullerene complexes. And finally, I elucidated the effect of microstructure on radical scavenging abilities and discovered that amphiphilic polymers in aqueous solution exhibits a double exponential trend, or two reactions. So what are the overall impacts of this project? I thought that there were sort of 5 prongs. So environmentally, first of all, NCC can help facilitate a move towards more environmentally friendly materials that are greener and cleaner in both production and disposal. Furthermore, cellulosic materials are an excellent carbon sink that help to trap carbon dioxide. Economically, I actually read a Globe and Mail article yesterday, and this is the quote I found, that NCC is a key strategic piece of an industry-wide attempt by Canada's embattled forestry industry to reinvent itself. Canada is already a world leader in the fields of research and production of NCC, I believe that this small nanoparticle can continue to help Canada become a key player in the future green economy. The third area, biotechnology, it has effects in biotechnology because NCC can actually be produced by bacteria, and the qualities of this NCC can actually be modified based on the conditions that the bacteria is grown in, which I found really interesting, and which I think is a really exciting different field of research. Health-wise, as I referred to the problem at the beginning of my presentation that I was attempting to solve, NCC can provide that stable biocompatible antioxidants and can be used as supporting therapy for diseases ranging from cancer to Alzheimer's, and also can be used as supporting treatment during other medical treatments like chemotherapy. And finally, in terms of lifestyle, NCC can be used as a nutraceutical to support health, or as a preservative in food, or even as a cosmetic ingredient that will help to reverse the effects of aging or smooth out those wrinkles my mom always complains about. I would like to thank Dr. Yao Zhao Ling for her help, and Dr. Liu Zhewen for the use of his equipment, Dr. Brad Bass for his advice, Sanofi for their financial support. I would like to say that I was really excited about this because there's no research, as far as I know, that has been reported on the antioxidant properties of NCC. And so I believe that this project represents an exciting new discovery that can open up a vast new field of possibilities for this renewable resource. Thank you for your time.
Thank you very much, Nell. Lots of exciting stuff. Questions? Jim?
Sure.
So you ended your presentation by saying there was no prior research on the antioxidant properties of NCC. Mm-hmm. So I guess what made you think about this? You know, why you think NCC would be a good antioxidant?
Mm-hmm. Well, I actually did a project in this last year in the same lab that I'm working in this year on drug delivery. NCC has been seen as more of a carrier for the drug and not actually the treatment itself. So last year I worked on it as a carrier for drugs into the body. But as I was talking about my project for this year, I was talking about it with my mentor, and she suggested, you know, the chemical structure suggests that it could possibly be the treatment itself. And so I decided to look into it. I found research that showed that hydroxyl groups are linked to antioxidant activity, and so decided to pursue this line of research.
Can I have a follow-up? Yeah, as a follow-up, so if you were to utilize NCC for internal use, how would you administer it? What would you think?
Well, it's actually been developed as a lotion, so you could, because it's a rod-like shape, so it can easily permeate through the skin and go into your skin that way. Also, I've observed that microcrystalline cellulose, which is like NCC but on a slightly bigger scale, is already present in a lot of vitamin tablets or supplement tablets, so you could also administer it orally.
Alison?
Can you describe a little bit more about your statistics? You had lots of good statistical data there, so kind of describe how you did them, number of trials, et cetera, et cetera.
Sure. So I used, for the error bars, I had standard deviation, and I ran each experiment in triplicate, and I took the standard deviation from there.
Can you comment on why I guess the implications of the PDMA C60 being much higher than the NCC in terms of inhibition, but lower in terms of the rate, I think?
Okay, that's a good question. What I would say here specifically why its inhibition activity is higher is possibly because the micelles it forms are smaller, so they're more, the fullerenes in the hydrophobic micellar core are more accessible. But it's one of those cases where the antioxidant works really slowly, but if you leave it for a long enough period of time, it can scavenge more radicals than the other antioxidants shown here.
So with the PDMA C60, did you invent that?
No, it's polyacrylic acid, and that was synthesized by another member of the lab that I worked in.
Okay, so where I was going, like, if lab invented it, have they patented that?
I'm not completely sure, but I do know that there are very similar polymer fullerene complexes that have been patented and that are used in the market today in cosmetics.
So you would see this used as a transdermal cream, for example?
Yeah, or oral supplement.
Or oral supplement.
Yeah.
So being part of this project team, to me, would lead to you to be part of an owner of this type of technology,
Yeah.
which is very cool.
Yeah, definitely.
Alan?
Yes, so I'd like to. Has there been any study on the bioavailability of the NCC once it's ingested? So you say that it's absorbed when taken orally, and how long until it's degraded? Do you have a sense, or has it been done? Do we have a sense of the half-life of the molecule once it's entered the bloodstream?
Well, I actually meant to show you all this. It's kind of, but it's just wood chip. This is actually nanocrystalline cellulose, and this is it in dispersion. And what I found was that NCC, as a solid and even in dispersion, is very stable. So it doesn't, it only degrades at very high heat, and like after some mechanical sort of mixing, I guess. So it is quite stable. I'm not completely sure about the half-life. It would probably, like, if you can't digest it, probably be passed through your system. So however long it takes to go through your digestive system, if you're taking it as an oral supplement, would be the half-life in that case.
So the syntheses that you did, were you able to do all of that organic chemistry yourself?
Well, the first part, I got the procedure from a paper, which I cited there. And the second phase was actually based on work that my mentor had completed.
Okay, and a question about your problem with DMSO. Could another way to handle that have been just to subtract that as a background? So do you think it's possible to keep using DMSO? I think your solution is wonderful, and that you've logically gone through and found a solution. But do you think you could have proceeded with DMSO and subtract the effect of DMSO as a background effect and come up with essentially the same data and the same statistical significance?
I don't think so, and the reason for this is that if you go back to the original slide, the radical scavenging effect of DMSO was not uniform over different experimental runs. So I would have to figure out what the radical scavenging ability of DMSO was for each separate run, which is difficult to do because they're mixed together. So it's difficult to differentiate which is due to DMSO and which is due to the antioxidant that I was using. That's why I couldn't go back and do that. If I would, it probably would have been a lot easier. But I just had to sort of start all over again because of that. And when I found that out, I had actually read a lot of studies, which is why I used DMSO in the first place, that use DMSO as a solvent. So I'm not really sure if that is a factor that other researchers have taken into account, which I thought was kind of interesting.
Marshall?
You commented on the mechanism of action of how the hydroxyl groups on NCC have antioxidation for the free radicals. So how does fullerene do that? Like, did you have any idea?
Yeah. So how free radicals are reduced is either by the donation of a proton, which is how NCC works, or the donation of an electron, which is how fullerenes work. So because there is an abundance of pi electrons on the surface of fullerenes, because it's not an aromatic compound, but it has aromatic rings on its surface, these pi electrons are donated to the radical and then reduce the radical to form an adduct.
Any other questions? Jim?
Here.
Yeah, I like the way you did the analysis
Thank you.
and identified or hypothesized originally there's unimers and micelles in your fullerene NCC mixtures. But where did you get the solution of just unimers, and why would you only have a solution with only unimers in?
Okay. Well, what I did was I did some literature research, and I found this paper that was actually, it said that if you mix PDMA, it's a really specific polymer, PDMA, which is one of the polymers I tested, fullerene complex, with PDMA, just the polymer, in a ratio of 1:4 at a pH of 8.5, you get a purely unimeric solution. And the reason for this is there's a charge transfer complex that causes the fullerenes to repel each other. And then once you add polymer into the solution, the polymer then wraps around the fullerenes, causing like these isolated unimers throughout. And that was what was the result that was reported in one of the papers. So I just prepared that solution and then tested it.
So the fullerene is essentially encapsulated by the...
Yeah, it's sort of wrapped around with the
NCC.
polymer to create like a unimer, but it's not, it's different from a micelle. Yeah.
So can the fullerene still have antioxidant properties even though it's surrounded by the polymer then?
Yeah, well, it's more like an aggregation that sort of separated the fullerenes from each other to form unimers, as opposed to them aggregating in a micelle.
Janelle,
Yeah.
did I understand you saying this was your second year on this project, or was the first year's
No.
slightly different?
Well, last year's was in drug delivery. It was basically completely different because I made transdermal bioadhesives, and I used NCC as the carrier for the drug. This year, I'm testing NCC as the treatment, as the antioxidant that can help reduce free radicals to a stable state.
I thought I heard that. So I'm actually a little surprised that given the nature of the product, that you're not looking at this much more commercial sense, so to say, in terms of transdermal delivery patches.
Okay, well, I feel like commercially that would definitely be the next step. Well, what I was hoping to do would be to definitely, I did a slight cost analysis. I know that NCC is getting a lot cheaper because of the production pilot plants that are being opened in Quebec and in Alberta. And so I know that. However, I probably need to do more toxicity studies to exactly determine the effect on the body. Probably clinical trials would be another next step, and also streamlining of the processes I use to synthesize the NCC fullerene complex. But in terms of transdermal bioadhesives, that's definitely a good idea, and maybe I could combine my two projects to figure out a product that incorporates NCC as both the carrier and the drug. That would be interesting.
Would you think that the intellectual property will have to play an important role if you want to commercialize on this in the future?
Yeah. Actually, the second part of this, which was this phase here, my professor or my mentor that I'm working under is hoping to patent that process. And I'm also definitely, once I do a little more research into this and confirm and streamline what I've done, I'm hoping to seek that intellectual property patent.
Well, thank you very much for coming.
Thank you very much.
Enjoy the rest of your day and see you tomorrow.
Sure, thank you very much.